So we must obtain them from diets. An adequate diet must contain these essential amino acids. Typically, they are supplied by meat and dairy products. So it means that this kind of essential amino acid, you can get them from meats, and you can also get them from dairy products. So essential amino acids help the body function and regulate neurotransmitters, chemicals in the brain that control mood and behavior. Yeah, of course, actually, some of these amino acids that are considered as essential amino acids, they are very, very important because some of them, they serve as a raw material for the synthesis of some of the neurotransmitters. And when we say neurotransmitters are the chemical substances that help to control the activities of the brain. And these activities of the brain include mood and behavior. So, like for example, we have some of these neurotransmitters. Example, we have like serotonin. Serotonin is a neurotransmitter, and is the precursor of this serotonin is glutamic acid. This glutamic acid is an amino acid that is actually responsible for controlling our mood. So sometimes we are happy because of the production of this serotonin, and sometimes we are angry because of the lack of this serotonin. So these essential amino acids, they are very, very important in controlling some of the activities in the brain. So that is why if sometimes the deficiency of these types of amino acid can lead to disease condition, can lead to disease complication. And then a lack of essential amino acids, you see, a lack of essential amino acid can cause emotional and physical difficulties. And at the end, it can lead to health disorder. So it means that if you are lacking this kind of essential amino acids, it can cause a lot of problems like emotional changes, physical difficulties, and also it may lead to the health disorder. In fact, this deficiency of amino acid or this deficiency of an essential amino acid may also lead to death. So I think this is clear. One, we should understand that amino acids, they are amino acids that our body cannot synthesize them. The reason is because we don't have enzymes that can synthesize these types of amino acids. That is why they are called essential amino acids. You can only get them from the foods that we are taking. And examples of the diets that contain these types of essential amino acids, we have meats, we have dairy products like milk. So that is why it's very, very important to at least be taking meat, if possible, daily. You may not take much, but at least take as little as you can take in a day, because it can cause meet off with the daily requirement of the diets. Then they can also have the body functions and regulates the productions of some neurotransmitters. And these neurotransmitters are important in controlling the mood and the behavior, and the deficiency of these essential amino acids may lead to problems. Okay, so now the next thing now we are going to look at, this is the list of essential and non-essential amino acids. So what is the difference between essential and non-essential amino acid? It's simple. When you said essential amino acid, as I explained, our body cannot synthesize them. You can only get them from diets. While non-essential amino acids are those amino acids that our body can produce them. So we call them non-essential amino acids because we have the enzymes, machinery that can synthesize them. We have examples of them here. We have alanine, we have aspartate, cysteine, glutamate, glutamine, glycine, proline, serine, tyrosine, and asparagine are all examples of non-essential amino acids. So these are some types of these amino acids, like essential amino acid with hydrogen R group, sorry, with hydrocarbon R groups. So if you ask which types of the amino acid that have hydrocarbon R groups, we have them here. We have isoleucine, they have hydrocarbon. What is hydrocarbon? From basic knowledge of organic chemistry, they are compounds that contain only carbon and hydrogen. So they only contain carbon and hydrogen. So examples of them, we have isoleucine. If you look at what we have here in this R side chain, to look at it, is carbon and hydrogen, carbon and hydrogen. So they are called essential amino acid with hydrocarbon chain. We have valine also. Look at it, valine is the other side chain, have only carbon and hydrogen. So that is why it's called essential amino acid with hydrocarbon R groups. Look at also leucine, it's the same thing. Look at also phenylalanine. This is the ring. And of course, it has something, carbon and hydrogen. And then the next thing that we are also going to look at is those essential amino acids with neutral R group. Those with neutral R group. We have tryptophan, threonine, and methionine. And those with acids or base R groups. So those with base R groups, we have histidine and arginine. They are the one with base group. And the one, yeah, of course, look at the lysine also. It has a base group. So we have these kinds of amino acids, their classification based on the types of the R side chain. I hope it's clear. So now the next thing that we are going to look at is Zwitterions. What is Zwitterions? When we said Zwitterions, it means that are the amino acids that have both positive and negative charge, but overall, their charge is always neutral. And there is also another name we give this Zwitterion. We call it internal salt. So although alpha amino acids are commonly written in the unionized form, this is the non-ionic form of an amino acid. Look at it here. This is non-ionic form of an amino acid. But what is Zwitterion? Zwitterion is the ionic form of an amino acid. So in the ionization case, the carboxylic group always have negative charge and the amino group have positive charge. So both the amino and the carboxylic group in an amino acid undergo ionization in water. They undergo ionization in water. So when they go ionization at a physiological pH, that is 7.4, there is a Zwitterion are usually formed. And what is these Zwitterions? It means that both the amino group and the carboxylic group will have positive and negative charge as reflected here. See, the amino group is having positive charge and the carboxylic group is having negative charge. So overall, you have positive and negative, so they will nullify each other. So as a result of that, overall, the Zwitterion will be neutral because the positive charge and the negative charge are neutralizing one another. So amphoteric, when you said amphoteric, amino group is protonated, while carboxylic group is deprotonated. Of course, remember from basic knowledge of acids and base that amino group or acid. What is an acid? An acid is a substance that donates proton. So because carboxylic group is an acid, so that is why we said it's deprotonated. When we said it deprotonated, it means that it gives out its proton, that is hydrogen. So look at it here. In this case, before the ionization, in the non-ionic form of the amino acid, you have OH group. But what happened after ionization? This hydrogen here is being removed. So it means that there is deprotonation of carboxylic group. So it gives out, or it donates its protons. And then the amino group is protonated. That is, the protons is added to the amino group. So that is what happened here. You see, in this case, if you look at it here, in this case, there is no proton. It means that the carboxylic group is deprotonated. And then the amino groups is protonated, because amino group reflects the basic nature of a substance, the base properties. And then the carboxylic group have the acidic properties. So as a result of that, there is deprotonation from carboxylic group and the protonation in the amino group. So, and one thing is they are soluble in polar solvent due to the ionic character. So generally, the Zwitterion form or the Zwitter-ionic form of an acid or of an amino acid is soluble in organic solvent. And the reason is due to the presence of the ionic character, that is the presence of both the positive charge and the negative charge. So ladies and gentlemen, a simple term, when we said Zwitter ions, it means that it's the ionic form of an amino acid. And in this ionic form of an amino acid, generally, they are overall neutral, because you have positive charge from the amino group and the negative charge from carboxylic. So they will nullify each other, and they are amphoteric. Why is that they are amphoteric? It means that they have some component that can interact with water, while some component will not interact with water. So now the next thing that we are going to look at is acid-base properties. Although I think there is something very important that I need to actually explain here. Generally, the ionic form or the Zwitter ionic nature of an amino acid change with a change in the pH of the environment. So if the pH is changed in the environment, there is a likelihood that the charge or the Zwitter ionic forms of an amino acid will also be affected. So you should understand this. The Zwitter ionic form of an amino acid is usually affected by the change in pH of the environment. So now the next thing now that we are going to look at is the acid-base properties of an amino acid. So look at it now at a pH of 1. The net charge of an amino acid is positive. And of course, look at it at a pH of 7. The net charge is 0. Why? Because there is both deprotonation of carboxylic acid and the protonation of an amino group. But when the pH changed to 13, there is only negative charge on the carboxylic group. So you see, actually, if you can now look at this figure, you will be able to understand that at the pH change, there is a likelihood that the charge, the amino acids, might also change. So this is what happened in the case of these amino acids. Okay, so now the next thing that we are going to do is the reactions of an amino acid. So amino acid undergoes a series of reactions. Amino acid can undergo a series of reactions, and these reactions involve the reaction of the carboxylic group and the amino group. So now we are going to look at the some of these characteristics, some of this reaction. So number 1, we have reactions due to carboxylic group. And examples of these reactions we have number one. It means that those reactions that are involved the use of the carboxylic group from the amino acid include A, salt formation with alkalis. So it means that if you have alkalis and it now react with the carboxylic group of an amino acid, a salt will be formed. Remember, this is just a basic neutralization reaction. And there is an ester formation. Of course, remember, ester formation is a reaction between carboxylic group and alkanol group, that is alcohol. So there is usually formation of ester. There's also amide formation with an amines, and there is also decarboxylation. So it means that when amine react with carboxylic group, usually amide formation, there is formation of amide group. And there is also decarboxylation, that is the removal of carbon dioxide from the amino group. So these are just a few reactions of the reactions due to carboxylic groups of the amino acid. Number one, neutralization. Number two, esterification reaction. Number three, we have amide formation. And then the last one, we have decarboxylation. Then there's also reactions due to amino groups, which include deamination and reaction with ninhydrin. So ninhydrin reagent usually react with amino group of amino acids to yield a color product. And the intensity of this blue color indicate the quantity of an amino acid. It means that the more the blue color, it means that the higher the quantity of the amino acids. So ninhydrin can react with an amino group as proline and hydroxyl group to gives a yellow color. So you see, in this case, ninhydrin can react with amino acids as proline and hydroxyl proline to gives a yellow color. So it might not necessarily to give always blue color. So now the next thing is detecting amino acid using ninhydrin.